You've probably heard about graphene. This material is obtained from graphite
and can be used to produce a variety of other materials.
It is quite interesting, especially in the biomedical field, because this material has a great potential in the development of biosensors, as drug delivery, tissue engineering, development of new strategies for bioimaging, and cancer treatment.
But graphene has important limitations, such as the high hydrophobicity, which can hinder the real applicability. However, the oxidized form of this material, known as a graphene oxide, is more hydrophilic and can be covalently functionalized by modifying the oxygen groups on its surface. This characteristic, combined with the high surface area of graphene oxide-based materials, makes them excellent candidates for drug delivery. Although many research groups have developed graphene oxide-based drug carriers,
none of these products have been approved by the FDA.
One of the problems with the use of graphene oxide is the knowledge gap regarding its biological properties during its internalization by cells, which can be affected by the particle size and by modifications on the material surface. In addition, different ways of obtaining the material can produce uniform dimensions,
which does not guarantee homogeneous samples, and this can also affect the internalization process by the cells. In this context, an interesting research studied the relationships between graphene oxide and human serum, as well as the size of the nanomaterial after this interaction in cancer cells and in non-cancerous lung cells. In the characterization study, 4 graphene oxide fractions were obtained, and their average diameters were determined by DLS and SEM images. As a result, the 4 fractions had different average diameters. Although DLS is not completely accurate for measuring non-spherical particles, it provides valuable information about the size distribution of an entire sample. After this data, the authors decided to use only GO2 and GO4 for the next experiment, because the significant difference in size between them were more noticeable, and the dimensions of these fractions correlated better with the expected size than GO1 and GO3. The structural changes in the initial graphene oxide suspension caused by the interaction between the samples incubated and non-incubated with a human serum were studied comparatively by Raman, FTIR, and elemental analysis. The intensity of the Raman spectra signals in the graphene oxide incubated with human serum was lower than that of the particles that was not incubated. This may have been caused by absorptions of protein from the serum to the surface of the nanomaterial. After the convolution of the spectrum, the authors conclude that contact with the serum components introduced more defects in the material structure. The FTIR spectrum of the non-biotransformed graphene oxide showed more peaks. This may also have been caused by the absorption of molecules on the particle surface. These finds were confirmed by elemental analysis, which showed that the biotransformed sample had much more nitrogen groups, which may indicate the absorption of proteins. For the toxicity experiments, biotransformed and non-biotransformed GO2 and GO4 fractions were incubated with the cells for 48 hours. Cellular metabolic activity was measured with the Alamar Blue assay at 3 different moments: before the 48 hours incubation, after the 48 hours incubation, and 24 hours after that. The results showed that the cells incubated with serum exhibited more metabolic activity. For the cancer cells, there was a reduction in metabolic activity in the large particles, or graphene oxide, which was not incubated with serum. Because of this reduction, for the next experiments, the authors chose to work only with the concentration of 50 micrograms per ml. Although the cancer cells showed a reduction in metabolic activity, the number of dead cells stained with propidium iodate was not as high as the author expected. This may seem strange, but the non-biotransformed GO4 could interfere with the cell cycle of these cells, causing a reduction in metabolism without killing them. Confocal laser scanning microscopy was used to determine the accumulation of graphene oxide in the cells. For this purpose, the particles were modified with FITC. The results showed that the 4 samples had a high affinity for both cells. The fluorescent graphene oxide was close to the cell nuclei, and the intensity of the fluorescence signal for graphene oxide was higher in the non-biotransformed samples. To better study the differences in the efficiency of cellular internalization, the average fluorescence intensity of the samples was measured using flow cytometry. Cells that were not exposed to graphene oxide were used as a negative control. The results showed that the larger particles GO4 entered the cells more efficiently in all cases for both cells. Furthermore, the internalization of graphene oxide was more efficient in the non-biotransformed samples. So in conclusion, in this study, we have some interesting results. The large particles were more efficiently internalized than the smaller ones, which contradicts many published studies. Furthermore, the biotransformed part was less internalized than the non-biotransformed one, which is also a contradiction with other published studies, including mine. The main reason for it is that biocorona can increase the cellular internalization of nanoparticles. The author said that more studies are needed to fully understand the biological behavior of graphene oxide in the entire biological system. So we can understand that we still have a lot of work to do, but the future looks promising.